We've been talking about cytoskeleton. This is the third introductory module on cytoskeleton. While it's a complicated topic, I'm going to take this opportunity and explain to you how a cytoskeleton is dynamic. It's not a fixed scaffold that does not have ability to change. Cytoskeleton reorganizes in response to different signals. We have talked about cell signaling. So let's see how signals can influence the architecture, the cytoskeletal architecture of the cell. Arrangement of cytoskeletal elements determine the shape of the cell. We have talked about it, and I have shown you the cage, the bird cage, and I have explained to you how cytoskeleton determines the shape of the cell and how the shape of a cell is very important in cellular functions, the functions a particular cell performs. In response to environmental cues, cell may need to change their shape or move. Changing shape or moving of a cell is critical for their function and or of course their survival. Change of cell shape would not be possible if cytoskeleton was a fixed element held together by permanent bonds. So in response to different environmental cues, cells have to change their shape, and shape is determined by the cytoskeleton which is present inside the cell. And if the cytoskeleton was a fixed structure, cells will not be able to change their shape or they would be able to move. So we have to have a strategy which will allow us to have structural support as well as flexibility to rearrange. You can do that if cytoskeleton is made up of smaller units which are held together by weak interactions rather than strong forces like covalent bonds. Cells undergo rapid structural reorganizations, disassembling filaments at one side and reassembling them on a different side. Let me show you graphically what I have just said. Here, for example, we have a cell. It receives a signal that there is a nutrient, for example, present at this side. The cytoskeletal elements, which were present here in form of polymer, now they can disassemble. Here on the slide, on the other side of the slide, you can see it's sort of a reversible reaction. The monomers, they can come together and form the polymers, and when needed, polymers can disassemble and form the monomers again. So first of all, cell has to depolymerize its skeletal elements. So these skeletal elements on this side will first of all depolymerize, and they'll become monomers again, as you can see in the next sequence. And now, in order to move in this direction, cell will have to extend its membrane. One of the ways cells can extend their membrane is by assembling skeletal elements, which will push the membrane in the desired direction. So when the skeletal elements start assembling here, forming polymers, they can push this membrane or the cell in this direction. Consequently, as a result of which, the cell will move in the desired direction. So you can also think of this as an analogy would be just like buildings are made up of smaller bricks. Buildings are very, very, very tall. Just like that, if we are drawing analogy, the monomers are a few hundred nanometers. They're in few nanometer size, whereas the structures they are forming are rather large. So again, we have this flexibility. Intermediate filaments are made up of smaller subunits that are themselves elongated fibers, whereas actin and microtubule filaments are small, are compact and globular. So I'll show you, of course, diagrams of monomers of both of them in more detail in later on. But for now, the point is actin intermediate filament monomers are long and filamentous, whereas microtubule and actin monomers are small and globular. All three cytoskeletal elements form helical assemblies of subunits which self-assemble using a combination of end-to-end or side-to-side protein contents. contacts. I'm sorry. So two ways. You have a protein here. It can have here, for example, are two subunits. They can form contacts. They can stabilize. The contacts could be lateral. This is a lateral contact, or it could be longitudinal, which is in this direction. These two are binding each other longitudinally, and these two are binding laterally. So assembly and disassembly can occur rapidly without the covalent bonds being formed or broken. So these are weak interactions that I've drawn on the screen. Differences in structures of subunit and strength of attractive forces between them produce critical differences in the stability and the mechanical properties of each type of filament. So these forces, the amount of forces between the subunits will determine the stability and mechanical properties of the filaments. So, for example, actins, as we will see, actin filaments have different mechanical properties as compared to microtubules. So these functions, or these elements, the skeletal elements, they interact with other proteins which have significant influence on their different properties that we have talked about. These proteins collectively can be called cytoskeletal associated accessory proteins, or CAAPs. They regulate the distribution in the three-dimensional space of the cell, the distribution of these filaments, cytoskeletal filaments, and also their dynamic behavior. So where a filament is going to depolymerize and where it is going to polymerize, in the case we saw earlier, there has to be something telling the cell to depolymerize its microtubule or skeletal array and then repolymerize at a different location. So that is a function that is performed by the cytoskeletal associated accessory proteins. Acting together, the accessory proteins enable a eukaryotic cell to maintain a highly organized but a flexible internal structure, and also in many cases move, as we saw in our example earlier. CAAPs bind to filaments or their subunits, and they can determine the following: the site of assembly of new filaments. They can regulate the partitioning of the polymer proteins between filaments and the subunits. They can change the kinetics of filament assembly and disassembly, harness energy to generate force. We will talk about motor proteins. They are also cytoskeletal associated accessory proteins. And also, they can link filaments to one another or to other cellular structures. For example, we saw when we were talking about cellular membranes, cell adhesion, how skeletal elements, how, for example, actin filaments or intermediate filaments anchor in specialized structures which are made up of different proteins. CAAPs also brings cytoskeletal structure under the control of extracellular or intracellular signals. So these are different functions that CAAPs perform in a cell, and we'll talk more about them in the later modules.